you so much for coming. It's a delight to see you all here today. I'd like to start by acknowledging the traditional owners of the land in which we're meeting today, the Turrbal and the Yagera people. I pay my respects to elders, past and present, and recognise their connection to country and their role in caring for and maintaining country over thousands of years. We're going to start by playing a video that talks a little bit about Cure for CF, and then I'll chat a little bit more.
I began Cure for CF because I saw an opportunity to help fund research to find a cure for CF. It was as simple as that.
Cure for CF has a single focus: to find a cure for the disease. We're not being distracted by anything else. The thing that we want to do is to find a cure, and all of our research dollars goes towards that.
Cure for CF is focused on translational research. And in its simplest form, and the easiest way to understand it, is research that can and hopefully will lead to a tangible outcome for the person living with the disease.
There's not a lot of time for people with CF. They do have a life expectancy that is less than half of that of the average normal lifespan, so we're in a hurry.
For me, being a CF warrior means that I can help find a cure and do my part in getting to that cure as soon as possible.
So since the foundation started in 2009, it's come a hell of a way. I think from memory we raised about $40,000 in that first year. To come 10 years later, we raised just on $1 million.
Today, Cure for CF is the country's largest private funder of CF research.
As long as we have the support from donors, from our sponsors, from all the people that fundraise and all the people that put their heads and hearts and talents to support our cause, we will be here fighting for a cure.
So by focusing on translational research and that laser-like focus on a cure, what it means for somebody living with cystic fibrosis and their families, it means hope. Hope that one day a simple pill or a jab will be able to rid them of this disease.
A cure would have an incredible impact on those families. And I know the impact that would have.
We may not be able to be together in person, but we are in spirit. That is how we will conquer this disease.
Cystic fibrosis is hard to beat, but with the army that we're creating and with the momentum that we're gaining, we know that CF will one day stand for Cure Found.
I'm all for the fight.
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 Are you all for the fight?
Thanks, everybody. Yes. From me, is that better? Could you hear the video okay? Perfect. No worries. So, as I mentioned earlier, we're delighted to be here in Brisbane today. We originate from South Australia. So it's been a couple of lovely, not so sunny days. I'm not sure whether we brought our really cold weather up with us, but as we leave on Thursday, I hear there's 25 in the sun. So hopefully we'll get a few rays before we leave. It's amazing to be surrounded by people that are all here for the same common goal, and that is to find a cure for cystic fibrosis. Cure for Cystic Fibrosis was established in 2009, and you saw on the video Greg, our patron now and our founder. He was part of a parent-led group that was really frustrated with the lack of cure-focused research that was happening across Australia and the lack of outcomes. And so he decided that he wanted to bring a group of people together to raise funds focused solely on cure-focused research. So research that has the best chance of making it into the hands of people that need it most. We are today, we raise funds across the country and we direct them to support promising CF medical research. You would have received a booklet as you came in, and that would give you a greater understanding of our grant program and our latest research projects. So what we do is we connect passionate people about finding a cure with leading CF researchers, and we're looking to find the smartest and shortest possible way to get a cure. As you would have seen, we have one focus. We only fund research, and we only fund life extending and cure focused research. So our goal is to see the research that we support translated into clinical benefit. We emphasise this from the very beginning, and we do this by prioritising projects that have a clear translational path and researchers who are committed to taking the necessary steps to see their innovations in the hands of patients. This is our point of difference. And at Cure for CF, we pride ourselves on transparency. Everything we do is for this cause, and you can clearly see in our annual reports how much we raise each year, how much we spend in running Cure for CF, and how much of the money that we raise is invested into research. I'm proud to say that Cure for CF is Australia's largest private cure-focused research organisation. Last year, we invested over $700,000 in new research, and our goal this year is to increase that amount to $800,000. As a small not-for-profit organisation, our superpower is our ability to surround ourselves with the brightest minds, both nationally and internationally. Our highly skilled and innovative voluntary board and committees, our passionate ambassadors, as well as our research partners, are all shining a spotlight on our cause and on our fight for a cure. This is supported by a robust community of fundraisers, corporate and pro bono partners, and generous donors. And it is this collective that will see us accelerate life-extending solutions for all people with CF. In a moment, our research development lead, Jodie Simpson, will talk a bit about how we're building a portfolio of research to attack CF on many fronts. But for now, we'll share another little short video that shows just how far we've come in our fight against CF. Thank you.
Bit of a hard act to follow. Hi everyone, I'm Jodie Simpson, the Research Development Lead at Cure for CF, and it's a great pleasure to be here tonight and talk to you a little bit about our research grant process and the kinds of research that we fund. So as Susie mentioned, our goal at Cure for CF is to fund research that has the potential to transform CF globally. We can only fund the best research if the best researchers apply, and that's why we're here in Brisbane for a few days. We're meeting with researchers, we're meeting with some of the CF teams, and we want to work with researchers who want to translate life-extending therapies to patients. Our grants process ensures our focus is on funding research that has scientific, clinical, and translational merit, and a focused pathway to clinical outcomes. We ask the applicants to tell us what the next step will be, how they will move their research closer to the hands of patients. We have a rigorous funding model and excellent governance and reporting frameworks, and we have an independent advisory committee, and this committee is made up from stakeholders across the scientific, clinical, translational, and consumer perspectives. We are looking everywhere for research, not only in the CF field, but we recognise that there is great research happening perhaps outside of CF that can be used to help transform lives. Some of our researchers funded a program that aimed to attack the deadly Burkholderia cepacia bacteria. Firstly, some time ago, we funded a project at the Walter and Eliza Hall Institute, and more recently, we funded some work at the University of Melbourne with Dr. Nicholas Scott, who is actually developing some vaccination technology in the hope that one day he'll be able to vaccinate people against that bacteria. The research we fund has impact. In 2018, we funded a phage project from the group at the Basil Hetzel Institute in South Australia, and they were investigating how to eliminate Staphylococcus aureus bacteria using new bacteriophage therapy. Following on from that funding from Cure for CF, the team were able to apply and receive a grant of more than $1 million from the Medical Research Future Fund. And they now have recently commenced recruiting for their clinical trial that's looking at applying bacteriophage therapy into the sinuses of people with that particular bacteria and infection. We use our CF community to promote this impactful research and let the community know what clinical trials are happening, and if they want to participate, they're able to connect with the researchers. So the other part of what we do is we want to connect our community with our researchers. Excitingly, we funded another phage project, which was mentioned in the video. This is a world first personalised treatment for children and young people with a particularly nasty and tricky bacteria called Pseudomonas, and that's being led by Dr. Dev Singh and his team at the Children's Hospital in Sydney. This work was recently featured on the project, and I think we're going to show you a little video about that. And I'll be quiet for a minute and then I'll come back and talk to you a little bit more.
It's just an infection in my lungs, and it just keeps me from doing some certain stuff that a normal person would do.
12-year-old Abby has cystic fibrosis, and like many with the condition, she's more prone to bacterial infection in her lungs.
We need to use strong antibiotics, and that course usually is about 10 to 14 days.
Dr. Dev Singh has been part of Abby's medical team for a decade, but the bacteria in his patients are becoming more resistant to antibiotics.
You know, the IV antibiotics aren't really working, so we've sort of got to try something else that's going to keep her healthier for the future.
For instance, bacteriophage. Should we give it a try to see if we can actually get rid of this bacteria in her chest?
Bacteriophages, you know, bacteria eaters, that's where the name comes from, are viruses that prey on bacteria. They're probably the most numerous and diverse life form on Earth.
Professor John Iredell is head of the Centre for Infectious Diseases and Microbiology at Westmead.
If your enemy's enemy is your friend, then bacterial viruses are your friend when you have an infection.
And finding these viruses is fairly simple.
So if you are worried about an infection that normally lives in the human gut, you look in the human gut or in sewage. If you are looking for a bug that has opportunistically got into a chest but normally lives in the soil or a river, you go to a river or the dirt to find it.
John's team match phages with superbugs. It's part of a global fight to control infections against which antibiotics are increasingly powerless.
Predictions are that within a generation, if the trajectory doesn't change, then it will be the leading cause of death, more than cancer, more than heart disease, #1 cause of death globally. So it's really a major health and economic emergency.
Once in your body, phages will attach to a targeted bacterial cell. They then inject their own gene material into the host. The phage replicates inside, eventually rupturing the cell. The new phages repeat the pattern, killing off the problem bacteria.
At the moment, the main demand is people who have just, really failed conventional therapy.
The team at Westmead has saved dozens of lives already by working with hospitals around the country. But this isn't a new science.
I think our Georgian and Polish colleagues precede us by many, many decades. It's a routine therapy, so they've got this enormous experience.
In Georgia, patients are regularly prescribed phage therapies instead of antibiotics.
And clearly there's a lot of different bacteria in the world that could cause an infection. That means you need to have a lot of different phages.
And the team at Westmead is leading international efforts to link up phage libraries.
We've shown that you can make a phage and send it anywhere in the world on a jet now, and it's fine when it gets there. Well, why not just connect all the repositories in the world so that we can just exchange them?
It was John's team who received Abby's bacteria from Dev.
And from there, we got about 6 bacteriophages that showed promising results, and we picked the best one for her.
Abby is the first patient in the phage trial. Every 6 months, she spends 2 weeks in hospital for treatment.
She usually comes in around about the 80% and the lung capacity gets up to about 100 when we leave after a tune-up.
When it's low, it's like hard to breathe and it feels like you're kind of stuck in one place and the lungs just feel really tight.
Abby has to travel 700 kilometres to get here from her home in Lightning Ridge in northwest New South Wales.
I work full-time and single mother, so yeah, for 2 weeks it's quite hectic.
While it's only the first of 3 phases of testing and nothing is official, the phages appear to be doing what they're supposed to do, killing the bacteria.
It's amazing that it's working, so we're pretty stoked to be able to have a go at it firsthand, I suppose.
Perfect.
Wonderful.
Eventually, over time, once we prove that it's safe, it is efficient, we want to see people living with CF being able to do this treatment even at home.
Wow.
It's really game-changing stuff, especially as someone as a person with disability. I was a young kid with disability. Just medical advances, mate.
Thank you. I always get excited when I see that video. And I caught up with Abby just a couple of weeks ago. She was down having her 6 monthly checkup and she's still Pseudomonas free and doing really well. So we're looking forward to keeping track of how she's progressing. And that study has just recently recruited its 3rd participant. So it's early stage clinical trial, but we're really excited to see what comes next for bacteriophages. So one of the most wonderful and rewarding parts of my work is to also be able to take some time and look at the CF research that's happening around the world. It's because it's important that we ensure that we're funding the best research. And we know the best research happens in Australia as well as in other countries, but we want to make sure we're funding research here that makes it to patients in Australia. Last year, we funded an exciting new project led by Dr. Elena Schneider-Futzschick from the University of Melbourne. And she's working on a project to try and understand how Trikafta, one of the modulator therapies, moves across the placenta during pregnancy. So this project is really going to bring new hope to both CF mums and CF babies. So Elena and her team their project is going to look at how that medicine moves from the mother into the baby. And she's really dedicated to understanding and learning what the right dose is to take during pregnancy, because while modulators are very effective, they haven't been tested for safety during pregnancy. And so many CF women who become pregnant then are faced with such a terrible decision of whether to continue with their treatment or stop it. So Elena's work is going to help understand that. But the other thing Elena's work is going to do is look at how that treatment, as it moves across the placenta to the womb, might impact on CF babies during pregnancy. And certainly some early reports from other laboratories suggest that it's possible that many of those early CF pathologies are actually can be improved if there's exposure during pregnancy. So we're excited to be part of that journey with Elena and her team and be looking at the way forward as we come into this modulator era of CF. We know that modulators aren't the answer for everyone with cystic fibrosis, and there'll be new challenges that come up. So it's important for us at Cure for CF that we're across emerging treatments and emerging challenges for patients. Finally, we also fund research that has the potential to stop CF at the very cause. We are very excited to fund gene therapy projects, and we funded 2 phases of research from a group in Sydney. And we also have Dr. Jared Keiko here with us tonight from the University of Newcastle. And in a little while, he's going to give us an update on his gene therapy project that he's been working on at the University of Newcastle and the Hunter Medical Research Institute. So we're excited to have Jared here tonight. And we know that research is the answer to be able to deliver life-extending solutions. So I'm going to change tact a little bit now, and I'm going to stop talking to you as much. But tonight I wanted to introduce 2 guests. So I'll ask Christy and Jared to come up and take a seat on the interrogation couches. So at Cure for CF, we also recognise the importance of surrounding ourselves with expertise, clinically, scientifically, and equally as important, lived experience. We're just grabbing a mic for Jared there. But tonight I'm delighted to introduce Christy Thomas to you, who is the consumer representative of our independent expert advisory committee. You might remember I said our grant process, we have this rigorous process where researchers apply, they get shortlisted, and then the shortlisted grants go to an independent panel. And Christy is one of our very valued members of that panel. And I thought we might just ask Christy and Jared some questions tonight. I'm just checking our mics are working. So Christy, I might start with you and ask you, can you tell us a little bit about why you partner with Cure for CF and volunteer your time in this way?
I would love to. That's a little bit noisy there. Okay. My name's Christy. I'm a mum to a little boy with cystic fibrosis. So why I'm involved is a question I could answer either with my head or with my heart. But if we start with my heart, anyone who's had that type of diagnosis in their family knows that there's a bit of a line drawn in the sand. You've got life before and life after. And I had to decide how I was going to move forward. And I became very determined to be positive, and to look forward to better days and to have hope. And better days I knew meant brilliant scientists, dedicated fundraisers, and one day a cure. But hope can be hard to come by when you are dealing with a degenerative condition. And I was having a bit of a rough day and confided in someone at Cure for CF what kind of a day I was having, and it was because my son had his first serious lung infection with Pseudomonas, which we've already spoken a little bit about. And next thing I know, I got this little card in the mail, and they sent me a little note just to encourage me, and hopefully my boy will pop up on the screen, but the note said, "We're all in this together." And that stuck with me, and it's something that these beautiful people who've been presenting to you tonight have reminded me time and time again, that I don't have to do it all alone, which is great because I really can't. I can't give my boy the future that I want for him. I can't certainly find a cure without any scientific research behind me, but I don't have to do it alone. We are all in this together, and I've been reminded that time and time again by the brilliant people that I've had the privilege of spending time with. So surrounding myself with great research has always been, it's been a real game changer in my life and how I parent and how I move forward through difficult days. If I answer with my head, I think that Cure for CF are an exceptional charity, and what they achieve with just 2 or 3 full-time equivalent staff is phenomenal. And I'm always reminded that my hope is well placed when I'm working with such amazing people.
Christy. And as I mentioned, everyone, you are current consumer representative on our IRAC committee, and I wondered if you might share with us from your perspective why the voice of consumers is so important in the decision process of what research is funded.
I do a little bit of health consumer work in a few different spaces. Obviously, this is the one I'm most passionate about, but there's a catchphrase that we always use, which is nothing about us without us. And while I don't have CF myself and I don't purport to understand that person's experience, I think the closest you can get is a family member or a doting parent. And it makes absolute sense that if decisions are being made about the CF community, that there's someone there who can speak up on their behalf. And I try and do that for people. I don't have a scientific research background, so the team brings together a great mix of skills, and I'm forever in awe of their brilliance and how well they understand good research. I think it would be really hard to get a poor application past that committee. They are incredibly savvy. But I'm also grateful that they value the contributions that I bring. So I try and imagine not only the impact on my child, but on anyone in the CF community. So for people who can't access CFTR modulators or have a rare mutation, that they're not left behind. For people who are older and might already be suffering with organ damage, are we investing in research that can help and support them? So I'm grateful, but not all that surprised that I'm welcomed and valued as a consumer representative. You heard that Cure for CF started as a parent-led organisation, and they've never strayed far from that. They're certainly not a big corporate. And while they've grown and changed, they've got great governance and management and all those things that you need in an organisation that you trust with money. They've never strayed far from having consumers as the heart and soul of what they do. And only funding that research that will really change our lives. So that's why I know that whether I'm volunteering my time or fundraising, it's time and money really well spent.
So much, Christy. I think that's such an important contribution that you make as well, having been a researcher and someone who worked in a laboratory and counted cells, and it's so important to have that perspective of, it's easy as a scientist to have a really interesting question.
Absolutely.
And to go down a rabbit hole and find a really interesting answer, but it's really important to know, well, how would this be delivered to a patient? How would this impact that patient? How, we might have this wonderful treatment, but how do we actually deliver it and things like that? So your voice is so important. So thank you.
I think it could be easy for scientists to get excited about lots of interesting questions. So I'm glad that we always come back to the ones that are actually impactful.
It's too easy.
So the kinds of things that I would raise at a meeting would be, how quickly might this help people?
And I think it's bringing all those voices together. So we're grateful. So our grant round for this year, Susie mentioned, we've got up to $800,000 in funding. It opened 2 weeks ago. We're very excited to see what new projects will come in this year. And there's a few people in the audience that I've already had some chats to and correspondence who we hope will submit an application. We're really proud to have this funding to invest in life-extending research in Australia, but we always have more projects than we have funds available, as with so much wonderful research happening in this country. But that's why we continue to strive to work with our community to ensure we continue to grow those funds. Tonight, as I mentioned, we've got Jared Keiko here from the University of Newcastle. And before I get you to come and present, Jared, I wondered if you might share with us, as a current grant holder, a little bit about your experience of working with Cure for CF as a research funding body.
So thanks, Jodie. My experiences with Cure for CF has been fantastic. I realise there's some people in the audience who've probably never seen a grant before. Count yourself lucky. And then there's other people that spend a lot of their life writing medical grants. So the process with Cure for CF was very different. It's not a sterile process. It's a very interactive process, and it's iterative, getting feedback from Jody about your grants, about your progress, about the development. And then I'd say the other real big difference working with Cure for CF compared to any other body that I've written a grant for or been successful in getting a grant with previously is that it is all about the families and the people with CF, and you get to experience this directly. So I've had families, people with CF come and do tours of my lab with some of the work that I'm going to show you today. We've had families in there and see how this happens in real life, in real time. So that's a really unique process that I've never seen with any other organisation. And then I've been to previous events as well where we meet some of the fundraisers for Cure CF, amazing people, some of them doing amazing things like marathons, raising money for family members like children, nieces, nephews that have CF.
I think we know our CF community love coming and visiting the labs and putting on their lab coats and having a walk around. And we were fortunate recently, we were able to go and visit the phage laboratory down in Sydney, which was really exciting. And I know in that project snippet, it said, you know, it was easy to get phages out of wastewater, but I probably beg to differ on that one. It's quite the challenge. And if you're interested in how they can, you know, the water that you flush down the toilet can turn into an amazing treatment, in a couple of weeks, we're going to have a really exciting blog coming out from one of the scientists in the phage lab who's going to walk us through exactly how you go from wastewater to a life-changing treatment. So look out for that on our webpage. But now, Jared, we'll hand over to you to give us an update on your current research. Leave that mic on. You can go. And you're going to have a sit down.
Good. Okay. Hopefully you can hear online as well. So thank you very much for the invitation to speak here today, everyone from the organising committee from Cure for CF. So I'm going to give you an update on the work we've been doing with Cure for CF. And then I'm also going to talk a bit about another aspect of work we're doing in my laboratory, which will probably be of interest for people here in this room today and online. So I'm going to talk to you about how we're aiming to improve treatment of CF by boosting the function of the gene that causes CF, that's the CFTR, and trying to do this long term, so minimize the dosing. Okay, so our gene therapy, if you like, or our RNA therapy to be more precise, what it does is it works to upregulate, increase this CFTR gene. And the way it does that is that it tries to directly target the CFTR gene and leave it turned on longer term. So when you have genes in your DNA, they are tightly coiled like balls around a string. And this prevents them from being turned on long term. So what our therapy tries to do is target the CFTR gene and open up those balls into more waves of string. And this allows the gene to be turned on, so upregulated, increased longer term. Okay, so our therapy turns on the gene, and we've seen the effect can last as long as a month with one dose. And we encapsulate the therapy. So in order to deliver a therapy, we need to put it inside something so it can get into the airways or any other body system and inside the cells. So there's different ways in which you can encapsulate a drug. The way we're doing it is something called exosomes. And these are our body's natural system of encapsulating therapies or any kind of biological product. So you've probably heard of lipid nanoparticles. This is what the COVID vaccine was wrapped up in. This is how the COVID vaccine was encapsulated. So that's a type of synthetic vesicle. So we're using the body's natural vesicles, these exosomes. So we use these exosomes, they package our drug and they can deliver it inside the lungs and inside cells and that turns the CFTR gene up and leaves it on longer term. Now, how do we test this? How can it be relevant to the clinic? What's the test that we use? So we use what are called CF patient organoids. So what's an organoid? This is a three-dimensional organ-like structure that is derived from stem cells from a patient. So you can take them out of the colon, out of the rectum specifically, or out of the airways, out of the lung. It's best to take them from the colon because this has the highest level of the CFTR gene. So what does this look like in real time? So we take a tiny biopsy, preferably from the rectum of patients. It's very painless, doesn't require sedation, takes about 5 minutes. We then digest up that biopsy and we start growing them out. And so what you're watching is a video down the microscope over 3 days. So it's a time-lapse video. And from this biopsy with this particular patient, we're growing out the organoids. Now, the beautiful thing about this is that the organoids recapitulate all the genetics of the patient and all the characteristics around their epigenetics as well. So this allows us to create a really nice model that's personalized to that patient. Okay, so what are we trying to do with the goal of this work? Well, 2 goals. One, we want to help people that are inadequately treated with modulators, but are eligible to modulators. And so I'll talk about that in a minute. And then we also want to help people who are not eligible for CFTR modulators. In Australia, it's roughly about 11%. Other countries can be as high as 15%. So for people that are eligible for CFTR modulators, sometimes we see fantastic responses, both in the clinic and with their organoids, and the 2 things match. Our average response with these lines is getting about 40% of the normal function back for with the treatment with the best of these modulators, which is Trikafta. However, these are all people with CF that are eligible for CFTR modulators. And you can see that some of their responses are well below the average. So this occurs both in the clinic and in these organoids. And so there are some of these people that have no response, and there are some of these people that have a response that's well below their population average. So 5, 10, 20%. So what our treatment is so far showing that it can do is that it can boost these responses by at least 50% on average. And so far we're looking at longer term so that we may only have to dose once per month. And then we're also looking at helping people who currently miss out on CFTR modulators. So people with rarer CFTR mutations. So we're seeing some efficacy in people with what are called class 1 mutations, missense, nonsense. And then there's some other CFTR variants which we can see that it's not going to work in, where there's people who are missing a huge chunk of the gene. So where are we up to now? So we're using these natural exosomes, the body's natural communication system, its natural delivery system between cells in order to develop an inhalable product. So we know that if you compare lipo, which is the synthetic lipid nanoparticles that we used with the COVID vaccine, compared to this red bar, which are exosomes, we know that we get much better penetration into the lungs and much better uptake into the cells that need to be treated. So we're working with a company called Ab Initio in Sydney at the moment, who's Australia's premier company in soft mist inhaler formulation. And we're working to develop this into a wafer that can actually deliver these exosomes as an inhalable treatment. And then next month, talking about the translational spectrum, Cure for CF is very focused on funding work that's translational. We're moving into our first GMP, which stands for Good Manufacturing Practice, run in Sydney. And so what we're going to be doing is we're going to be scaling up the production of this therapeutic candidate to see if we can do this at much larger scale. Now we have to use GMP because GMP is what's suitable for clinical use, what's suitable for going into clinical trials. So we have to show that we could potentially do this. So next month, we're doing our first bioreactor run, which is where we're going to grow up lots of these exosomes in huge amounts in the New South Wales biologics facility, which is funded by the state government. Okay, so just for a few minutes, I'll switch to talking about some of our other work, which I think is probably of significant relevance for this audience as well. So this is a clinical trial called Origin 1, which stands for Organoid Guided N of 1. And all of this work is with Professor Peter Walk, who's the head of adult CF at the Alfred and formerly in Newcastle. So this is a clinical trial for people with CF that don't have the Delta F508. So to explain the background of this clinical trial, we took these patient biopsies, we grew up these organoids, and we tested the patients with these drugs in our what we call a CFTR test. It doesn't matter about the technical details for the purpose of this, but we test them with different drugs, and we predict with this CFTR test, do we think that they'll be a responder to different drugs or a non-responder? So here's two examples of particular people here. One person was predicted to respond really well to Trikafta, and they did. Another individual predicted not to respond to Trikafta, and they didn't. Both patients and all the patients in this trial were eligible for modulators. We followed the patients in the clinic and we looked at how they did in terms of their improving their lung function, their ability to breathe, and the number of exacerbations that require those antibiotics or hospitalizations. So overall, the population, as we know, Trikafta's on the market because this is the case. Overall, the population does really well, but it's a mixed response. It's what we call heterogeneous. Some do really, really well, some do okay, and some don't have any response. It's just showing you that. What we found was that our organoid test, our patient organoid test, was highly accurate at predicting what would actually happen in the clinic when they actually went on the therapy later on. So because of this, we've now moved into a clinical trial. So this is Origin 1. So as I said, we're trying to help people with CF that do not have the Delta F508 and are not eligible for Trikafta. So our idea is this personalized medicine, this precision medicine, we can use this test to identify people who may benefit from getting access to this therapy. And so we do the test, we identify the people that could benefit. I don't even have to go over that. Here's two examples of individuals with CF, both of them not eligible for modulators. We can see, unfortunately, one of the patients did not respond to any of the modulators, but another individual with this particular set of variants did respond, in particular to the one in purple, Trikafta. So this person's been enrolled in the trial. This is just their testing response. And we saw that they got a 23% increase in their CFTR function predicted by this test. They're now enrolled in the clinical trial and they're going to receive Trikafta. So our first cohort's already gone into the treatment arm and we're currently recruiting our second. So I'd just like to thank Cure for CF for all the support, all the wonderful things they do for the community and for inviting me to talk this evening.
Just invite you and Christy to come back and have a seat because we've just got some time at the end now to answer any questions. And so if you are joining us online and you have questions, Birgit's moderating the online webinar. You can pop your questions into the chat. But if you have any questions here in the room, Jess is here and I'm going to give her the microphone and she'll be able to bring that to you and answer any questions. And I might kick off and ask Jared a question. Got so many microphones. So it was really exciting to get that update, Jared. And I guess the first question is about the gene therapy project. And I love what you showed us about you're using a natural based packaging system, I guess, and now going down to Sydney and hoping that it'll be a wafer in like some kind of a puffer or something. Is that, what are you thinking it might look like for a patient?
Yeah, so we're testing different formulations. So the soft mist formulation, we're looking at different nebulization devices. So we're testing, I think, 9 different potential permutations of how it might best be done. We don't know yet. This is a first. No one's ever done this before, but it has been done with in other diseases with synthetic polymers. So we're pretty confident that one of these is going to work.
Fantastic. It's really exciting. Are there any questions from our audience, either about research or about the grants process or about Cure for CF? Yeah, oh, there's some up here. Tim and Matt had their hands up quickly.
I guess mine was just more of a scientific question, actually, about the mechanism involved. So, in terms of the boosting of CFTR expression, is this a general effect that happens across a whole suite of different genes, or is there some specificity to the control of CFTR?
So I left out a bit of the scientific detail there. Yeah. So we use zinc finger fusion proteins.
Right, okay.
And so we can make them highly specific to any gene, in this case it's CFTR.
Yep. Great.
Yeah, Tim.
Mine's non-scientific. So you mentioned the advisory committee that you'd be working with, and you said they had a lot of expertise. What sort of expertise is part of that committee?
So they're outside of our Cure for CF staff team, we have a research and commercialisation committee, and that's made up of some of our board members, also CF clinicians and other experts in, I guess, research and commercialisation. And then we have the independent committee, which has, I guess, some of the members at the moment, and you can see them on our website, but we have experts in research, in CF clinicians again. We also have people who've come from backgrounds in commercialisation and cancer, clinical trials, our fantastic consumer voices. And we have representative from Brandon Capital, and so other organisations like that kind of bring lots of perspectives to the table. So it's quite, as I'm sure, Tim, you know, and I know from having applied for grants over a long time, it's quite an unusual mix. But what's really exciting is it helps, we think, you know, bring the best projects forward. And I think one thing we have done is, you know, if people apply and they may be not successful, is we have people come back and take on that feedback and come back. And Dev Singh was one of those with the Phage project who he went back, worked for another year and then came back and was funded for that first clinical trial. So I guess, as you were saying, Jared, we kind of want to have that longer term relationship with our researchers.
That's unique.
It is unique. I know it's one of the things I love about it is getting, you know, Bigot and I work a lot with the researchers and catch up with them in between the official reporting period, and it's really a special part of our jobs. And connecting them, I think, is the other thing, because as researchers, we often work in different states and in different labs, and if we all can come together, I think that's a powerful opportunity to bring a cure or life-extending options forward for people with CF.
And the beautiful thing too is that these committees are all voluntary. So they're there because they want to be there, but they're really interested in the science too. And they're made up of some of your peers. And as we mentioned, Christy with her lived insights as well. But it's an amazing experience for us at Cure for CF to be surrounded by really, really bright minds and leaders in the field. And we get excited about everything that scientists say. We think everything that you guys say is amazing. So it's good to have people within the field that tell us, what's amazing and what's probably just really good. So it is we think it's a superpower that we have to have this committee as a voluntary committee, but also that cares so much about helping us to ultimately translate the work that we support.
I have a question from online. The question is, can organoids be taken at any centre in Australia for the trial, or is it necessary to come to Newcastle for it?
So we're doing both. It depends on whether the centre has ethics approval to do it. So far, the majority of the patients who tested, we have brought into Newcastle because it was more efficient to do that. But if a centre has ethics, then yeah, it can be done.
So if people wanted to know more about that, Jared, can we connect them with you if they say
they can contact you and then connect with me, and the study coordinator can give them all the details.
I can connect them with the study coordinator. So if anyone wants to know more about that, we can do that. And so you have had people actually come to Newcastle and have organoids collected?
Lots.
Exciting.
Yeah, many.
Any other burning questions? Or not burning questions? Yeah, go.
Just in terms of the grant scheme and the different criteria, I think they're all sort of weighted equally. But I mean, I guess I would imagine that sort of path to translation would be a big factor in considering these applications.
It is, and this year will be my third grant round I've been involved in, and what's been interesting kind of observing that has been that researchers often, when I see the answers to that part, I think sometimes panic and because we're not, well, it's a hard thing to know what's going to come next. We're passionate about this question and getting the right science. But it's really about having a plan so not just doing one project in isolation, about knowing what might come next or having the connections and the team around you that you might need to take the next step. And so I think it's more about the plan than it is about we've got all the IP set up and we've got a company. It's not, some projects that isn't the next step. And the committee knows that. So yeah, if that's helpful.
Too, it's about having the end user in mind. And when you look at where your funding comes from, organisations like Cure for CF are funded by parents and funded by the community. And so I guess their expectations are different and they want to see products in the hands of their children and their family members. So what's important is that the researchers that we work with have the right intent. You don't have to have all the answers, because the beauty is that the committees that we surround ourselves with, they also offer mentorship opportunities. And that's something else that sets us apart from other grant organisations, is that once you become part of the Cure for CF family, we want to make sure that you succeed. And so we will do everything we can to help you get there. So we've often had researchers that will give us a call and have some questions around the translation plan, and can we put them together with some of the experts that form our panel. And so we've been able to do that, and they're very willing to do that too. And they just help you to probably think in ways that you might not necessarily be thinking in, and to get sort of to get the ball rolling.
Yeah, great. Thanks for that.
Thank you. All quiet.
All quiet. Any other questions online?
I think we're right.
I think that's
Move
probably we're coming to the end of this part. I don't think we've got any other presentations. So what we'd love to do is, one, say thank you so much for coming out on a Tuesday night. It's Tuesday, isn't it? On a Tuesday night, and spending your time with us. It means a lot to us that you've come here to listen to us. We hope that it encourages you to get involved with Cure for CF, whether you're here as a researcher, whether you're here as a fundraiser, whether you're here as a parent, that in some way that you will connect with us, because Cure for CF is made up of the people who support it. And it really is your foundation to be funded by, but also to bring about life extension and cures. And we can keep going for as long as we have people applying for grants and the money to pay for those grants. So we hope that you'll engage in some way. For those people that are here in person, we'd love you to join us out in the atrium to get to know us a little bit better, but we'd love to get to know you a little bit better too. So we invite you to come and have something to eat and drink with us. Thanks for coming.